Dear audience, now I would like to talk about our research in the topic of computational immunology. Acne vulgaris is a common inflammatory skin disease, especially among adolescents. The acne lesions most often can be found on the face, but it can involve the neck, the chest, the shoulders, and other body parts. If the follicles of the healthy skin is clogged for some reason, the wax cannot enter the follicle, it gathers inside, and forms a good condition for the Propionibacterium acnes. After some time, the wall of the follicle ruptures, the wax and the bacteria reaches the nearby tissue, whereon our immune system reacts with an inflammation, which causes the clinical symptoms of acne. There are several factors that have been described in connection with the pathogenesis of the acne. For example, our environment, our age, our race, and the formerly mentioned Propionibacterium acnes bacteria. This bacteria is the member of the normal skin flora. It can be found on the skin of healthy individuals. It is known that this bacteria can be found in acne lesions, but its exact role in the pathogenesis is not well described. The presence of the bacteria helps in the rupture of the follicle. It activates the complement system, recruits granulocytes, and via antigen presentation, it activates our adaptive immune system, which sharpens the inflammatory response. In our results, we were examining the connection between the adaptive recognition and the pathogenesis of the disease. First of all, let's take a look how this antigen presentation works. These antigen-presenting cells ingest the bacteria and then digest their proteins into epitopes. After that, a molecule called MHC2 binds to this peptide fragment, and this whole complex is moved to the outer membrane of the cell. After that, this antigen-presenting cell travels to the lymph nodes, where it presents this epitope to the T immune cells. After that, the immune system knows what kind of cells should it attack. These MHC2 molecules have different specificity. Some kind of peptide fragments can't, others can bind to it with high effectivity. This binding depends on the amino acid sequence of the MHC2 molecule. This sequence is coded on our DNA, and it has thousands of variants. These variants are called the human leukocyte antigen 2 alleles. In our results, in our research, we were curious about if the different MHC2 molecules recognize the bacteria differently. If they do, does it end up in a different recognition in the worst populations? And if the population and if the recognition is different between the populations, does it correlate with the prevalence of the acne vulgaris? During our studies, we were examining the whole proteome of the Propionibacterium acnes. On this slide, you can see one protein. This is the ID of the protein, and this is the amino acid sequence of the protein. These MHC2 molecules can recognize 15-length peptide fragments on average. The method we use, using machine learning, examines the first 15 amino acid length phase and assigns a binding value to it. The machine then skips to the next 15 length phase and assigns a value, and the next, and the next, and the next, till the whole proteome is scanned. The phases with a binding value lower than 20 are accepted as potential epitopes. With this method, we examined 654 HLA2 alleles. Here you can see a part of the full proteome, and the highlighted place are the potential epitopes. If we combine these two data, we get a percentage which is specific to the studied allele, and it means its protein recognition ability. After that, we were curious about how the recognition of Propionibacterium acnes occurs all over the world. For this, we used the allele frequencies database, which contains the allele composition of different populations from all over the world. If we know what kind of alleles can be found in one population, and we know how those alleles recognize the bacteria, we can calculate an average proteome recognition ratio for one population. We did this calculation to approximately 700 populations. On this map, the circles are the populations, and the proteome recognition ratio is indicated with the color. The most intensive red means that some kind of populations in the world can recognize about up to 12% of the full proteome of the bacteria. As you can see, the recognition of the Propionibacterium acnes differ in countries. For example, in Indonesia and Croatia, the recognition is significantly higher than the average, and the lowest recognition we found in Papua New Guinea. As far as we know, the prevalence of acne vulgaris is different between races. So our next slide is really important. You can see the Austronesian race recognizes the proteome significantly higher than the American Indian, the Arabian, the Asian, and the Melanesian race. Well, at this point, we thought that we actually proved that people are different. It's not a big result. So we wanted to see if it's correlated with the prevalence of acne. For this, we were looking for epidemiological data. First of all, we found some really extreme data published about acne. For example, in Amazonia, where we found that the recognition is low, acne is a really rare disease, even in adolescents. In Papua New Guinea, where the recognition was the lowest, a study involving 2,000 patients haven't found a single patient with the symptoms of acne. After that, we were looking for articles which comparing the prevalence of acne between races. We found 4 good articles comparing 7 races. On the horizontal axis, you can find the recognition ability of the race, and on the vertical axis, there is the prevalence of acne in this race. As you can see, in those populations where the recognition is high, the acne is a really common disease. Where the recognition is low, the acne is a rare disease. Our strong correlational data validate our results, we think. So finally, let's see what can be in the background of this thing. As I formerly mentioned, the exact role of the bacteria in the pathogenesis is not well described. During the pathogenesis, the follicle is clogged. There is a huge mass of wax and bacteria within the follicle. After that, the follicle ruptures, and the bacteria reaches the nearby tissue. Based on our results, we think that those people who recognize the bacteria stronger, their immune system reacts with a more intensive inflammation to the presence of the bacteria, so they have a higher chance to get acne. Thank you for your attention.